Okay.
So we are now recording, and thank you so much, Patricia, for agreeing to give a talk, and it's all yours.
Okay, so hello, I'm Patricia Bassereau. So I'm currently in Paris, and I'm a director de recherche CNRS, so it's about like a professor in France. And I am working, as you can see here, I'm working at the Curie Institute. It's in the middle of Paris. So I'm not Marie Curie, you can see it's her. And this is the historical building of the Curie Institute. And as you can see here, we are located here, this is a river. So this is Notre Dame, and we are here at the Curie Institute. So I am by training a soft matter scientist and physicist, but in the middle of the 90s, I started to work on a biological problem related to membranes. And this is my group. So you can see this is a group in June. So we were back to the lab, but wearing masks. And the name of my group is Membrane and Cellular Function. And the objective that we have is usually to find out about mechanisms which are related to cellular processes, which involve membrane and proteins. And we have three main, let's say, topics in the lab. We work on cellular membrane organization and dynamics. We have also a large part of our project which is related to intracellular trafficking, endocytosis, exocytosis, and also some cell infection, and also some projects related to the formation of cellular protrusions like Filopodia. And you will see at the end of my talk what we do on that. And usually the approach we have is we collaborate with many cell biologists, microbiologists in Paris most of the time, but not only. And from them we get some questions. And we try to come up with an hypothesis, and what we do, and you will see that today, we reconstitute some of the questions that we want to address using in vitro system. And you will see this liposome or this membrane nanotube that we pull from liposomes. So we reconstitute the question, and then you will see how important is kind of to rebuild these functions. And we also collaborate with many theoreticians. So we try to come up with some theoretical model that can test quantitatively. And when we have that, we came up with a mechanism, and eventually we came back to cell biology. So this is let's say the general approach that we have. And in some cases, we work in life systems, and we work on cells mechanics. So let me get rid of that because it's really annoying, but anyway. So you see the full screen? Yeah?
Yes,
okay.
We see the full screen.
Okay. So as I say, the general question we address are usually related to cell membrane, and I'm sure that you know that not only the cell is surrounded by a membrane called the plasma membrane, but most of the compartment in the cell, like the nucleus, the ER, the Golgi, the mitochondria, they are surrounded by membranes, sometimes one, sometimes two. And the current view about the cell plasma membranes around the cell is that is given by this cartoon. So what the main property of I would say of a membrane is that it's fluid, meaning that the proteins which are here in the membrane, they can diffuse like in a liquid. But in addition, it's not only a fluid, but a fluid which can be deformed in the Z direction. So it can be deformed like let's say, yeah, it can be deformed. So in addition, the membranes contain, as you probably know, many lipids, more than 10,000 types of lipids, and many proteins. So some of them are included in the membrane, as you can see like this one, but some of them are bound to the membrane. And as I said, the membrane is fluid, meaning that the protein can diffuse. But as you can see here, there is also actin filaments which are bound to the plasma membrane, which is called the cortex. And being bound to the plasma membrane, this limits the diffusion of the molecule in the plane of the membrane. So there is not only actin filament, there is also other filaments, but it's mainly actin, which is involved in, let's say, confinement of protein in the plasma membrane. So if you want to build up a cellular membrane, so it means that you have to take into account all these components, the proteins, the cortex, the complexity of the membrane as well in terms of membrane of proteins and lipids. So a very convenient system to build up, let's say, a membrane system is giant liposomes. So a liposome is made usually of a single lipid bilayer, and it can have different sizes. But if you want to build up a system which has the same size as a cell, what you try to build up is what is called giant unilamellar vesicle, GUVs, and it's called giant because the size is of the order of tenths of microns of diameter. But locally, it's made of a single bilayer, which is about 5 nanometers. So if you want to build up something which mimics one of the cell membrane, you need I would say you need to be able to reconstruct different properties. And I would say it's like you would like to build up different modules for your membrane. So you have to take into account the complexity of the membrane, and I will start with that, explaining how it's possible to build up some membrane complexity, so meaning in terms of lipid composition, but as well membrane asymmetry, lipid composition asymmetry. You need also to be able to reconstitute membrane with protein, which are bound to it or incorporated into it, like this integral protein. And as well, you need to incorporate something which looks like the actin cortex or actin cytoskeleton. So you have to build up different modules. And over the years, I would say many groups have worked at building up some of the modules separately. And now what they try to do is kind of combine all of them to have something which has the complexity of a plasma membrane. So I will start with building up at least the lipid aspects of the membrane. So over the years, people have been interested to study what happens if you have a lipid mixing. So you take different type of lipids and you mix them. So how they behave? And they, of course, like in soft matter, they have been able to determine many phase diagrams. So what this phase diagram tells you here we have a mixture of 3 lipids. One is 𝗕𝘂𝗶𝗺𝗶, is unsaturated lipid, a sphingolipid, which is saturated, and cholesterol. So this is a mixture which kind of more or less mimic the composition of the basic composition of the plasma membrane. And if you look here, you have different domains in this phase diagram, which represent the type of organization that you have when you have some mixing of a different component. So you have different phases, which have different names, and I don't want to go in this now, but you can have this LD phase, which is disordered, liquid ordered phase, and also, and when you have almost no cholesterol and a lot of this sphingolipid, you can even have a solid phase, a gel phase. So people have been working a lot to determine the phase diagram depending on the lipid mixture they have been using. And there was a lot of also discussion, because at the same time in biology, people have found out that there is also some nano cluster, nano domains in the cellular membrane. And I would say that this created a little bit of ambiguity, because this system was made at full equilibrium, whereas in cell there is many processes which are not at equilibrium, which can also perturb this phase diagram. But at least people have been able to prepare vesicle with a very complex membrane. And here, this is an example where you have one of these ternary mixture where you have 2 lipids plus cholesterol, and they have fluorescent marker. And you can see that with this fluorescent marker, which segregates specifically in one type of domain, you can see that it was possible to identify different domain in this kind of giant vesicle. And here, this is a coexistence between these two fluid phases, the liquid disordered phase and the liquid ordered phase. And here you can see a case where you are more or less in this part of the phase diagram. And you have this domain which are not anymore, and which is a sign that you can have a gel phase. So this is typically which has been done by many groups over the, let's say, the 10, 15, 20 years. But in a cell, the membrane is not symmetric. So all these vesicles have been prepared with lipid mixture, which have a symmetrical distribution between the two leaflets. But in a cell, it's very well known that the membrane is not symmetric. And the lipid composition, let's say, the plasma membrane is different on the outer leaflet as compared to the internal leaflet. So this is here, this is a simulation done by Marink, which represents the lipid distribution between the two leaflets of the lipid bilayer. And you can see here that this is a representation of the head group between the outer and the inner leaflet. And you can see the outer leaflet contain a lot of sphingolipids, glycolipids, but no, and cholesterol. But none of these PI lipid or PS lipid, which are charged lipid. They are more at the internal leaflet, and you can see also a lot of P is also present. So you have a lot of this asymmetry in terms of head group, and as well, you can see also in terms of chain saturation or insaturation. So the question is, how is it possible to prepare membrane with asymmetric bilayers? Because all these giant vesicles I presented before are normally prepared from a lipid mixture that you dry and you rehydrate with different methods, and you eventually end up with a system which is perfectly symmetric. So there are different systems which have been developed by, for instance, in London, which has been very efficient in this field. So, and this is, if you are interested, this is a reference that gives you the method to prepare, one of the methods to prepare this asymmetric membrane. So one of them is to use cyclodextrin, which is a lipid transporter in a sense. And if you adopt this cyclodextrin with a lipid, which is you want to incorporate into the outer leaflet, so this leads to an exchange between the lipid of the outer leaflet with the lipid that you want to incorporate into it. So you can do this with a different type of lipid, so the saturated lipid or unsaturated lipid. And at the end of the day, you change the equilibrium between the inner leaflet and the outer leaflet, and you end up with a asymmetric distribution. So this is a technique which is quite efficient. Another technique which has been also developed by Gary Fagenson consists in using vesicles of a vesicle with a given composition and a supported lipid bilayer on a substrate, and is using calcium to induce the infusion of the giant vesicle with a supported bilayer, and this infusion leads to the exchange of the external leaflet of the GUV, and what you get at the end, this is if you rinse your calcium, you detach the vesicle, and you can collect a vesicle which have an asymmetric distribution. So this is a technique which are normally not used in oil, but there are also some other techniques which are, let's say, based on droplets. So water in oil droplets with some lipids at the interface. So a very efficient one is based on inverted emulsion, which are four. So this is an emulsion where you have a droplet with one type of composition, and you force a droplet by centrifugation or sedimentation to cross a monolayer, which has an interface between water and oil, which has a different composition. And what you do, by forcing these droplets through the monolayer, you end up by a composite membrane with two different leaflets. And this has been even pushed further by Manu Karian, who developed a technique, which is this technique with microfluidic, in order to get droplets which have a controlled size. And this technique is called CDICE. So this is using centrifugation and a monolayer, but you can also force two droplets covered with different monolayer of lipids. You can force them to lay together. And then at the interface, you have a bilayer, and this bilayer also is eventually made of two different leaflets. And this technique has been developed by �𝗂𝗵𝗱𝗶𝗂. So you see there is many techniques which are now available to produce this kind of asymmetric bilayer. But I would say the problem of this droplet system is that since it uses water in oil droplet originally, you have a risk to get some oil which is trapped into the bilayer at the end. So you can have some oil contamination, which could be a problem depending what you want to do with your bilayer. So this is what can be done, at least in terms of lipids only. So the very important aspect is to introduce proteins, and there are different types of protein, as I mentioned before. So I will start with the protein which are shaping membrane. And why is it so important? Because if you see this movie where you can see the Golgi apparatus in the cell, and you can see plenty of tiny points which move in the cell, which are vesicle, which are transported and form at the Golgi membrane and then transported into the cell by molecular motor. So it means that at some point this vesicle has to be formed from the Golgi membrane and detached. Same thing when you care about endocytosis. So in all these processes which involve traffic, what you have to do is to form the vesicle, you need to bend the membrane to sort the component which have to be transported, and eventually you have to cut the membrane. And here we have images which are related to clathrin coat mediated endocytosis. And here this is a corresponding cartoon. So here what we have is the beginning of the process at the plasma membrane, and we have the clathrin coat which is starts to form, and you can see that gradually when the coat is forming and many proteins are recruited at the neck here of the bud, you can see that the membrane is more and more curved here. And at the same time, you have the neck, which is more and more pinched. And at the end of the day, you have an important process, which is the fission. And the fission is performed by dynamine in the case of clathrin. And you have many proteins also, which are present here, including actin. So at that stage here, has been recognized that there are different proteins which have a very characteristic shape, as a banana shape, which are involved and recruited at the neck of the bud here, in addition of actin. And there was a lot of work which has been done to understand why these proteins are recruited at that stage and what their action when they are recruited on the membrane. So over the years, I wanted to show you also some work which has been done also on the codes themselves, so the clathrin here. So there was some work done by Aurélien Roux, who has been able to form, as you can see here, some structure which mimic the clathrin coat at the surface of GUV. And this is another example here of another coat which formed at the Golgi, which is called COP1. And you can see that also using the minimal component of the coat, it was possible to reconstitute some structure around GUV, which looks like some like buds mimicking a coat. So if now we go back to this protein which are found at the neck of the clathrin vesicle, this bar, they are called bar domain with this kind of structure. So there is, as I said, there are a lot of work which has been done to see what happens if you take a membrane and you incubate your GUV with this protein. So you can incubate them outside, you can encapsulate them inside, and you can see what is going on. And you can see here that if this protein, which have a very curved shape, like this protein is called amphiphysin and is found at the late stage of endocytosis in clathrin, you can see that they spontaneously tubulate the vesicle. The vesicle was originally smooth, and you can see plenty of these tubules around. This is another case here where you have another type, another bar domain protein, but you can see here that its shape is completely inverted compared to this one. And when you incubate a vesicle with this protein, you can see that you have a spontaneous formation of tubules, this case toward the interior part of the vesicle. And something also which is related more to cell pathogen interaction. This is a case where here we have a toxin interacting with a membrane. So this toxin is called Shiga toxin. Is a toxin which is normally secreted by a Shigella. And you can see that this, if you incubate a GUV, which contain the lipid receptor of this protein, you can see that the membrane spontaneously also form tubules. And this could be explained because when you can see on this simulation here, when the protein binds the membrane, you can see that there is a small deformation which is induced by this protein on the membrane. So it's not only this kind of experiment which tends to appreciate how much deformation you can have when you bind protein to membrane, but you can also be more quantitative. And we have been contributing to that by developing an assay based on membrane nanotubes to quantify how much a protein can deform membrane or sense curvature in a sense. And with this nanotube here, you can see that we have a GUV, and we pull a tiny tube from the GUV, which can have a diameter which can range between, let's say, 100 nanometer down to 10 nanometer. And with this kind of experiments, we have been able to show that the protein here, which is amphiphysine, when the tube is pulled, you can see here that we have a strong, so you see the tube is very green, as compared, we have here the lipid which are red and the protein which are green, and you can see that the tube is very green, meaning that we can have an enrichment of a protein on the curved membrane. And with this kind of a protein, with this kind of assay, we have been able to, over the years, to quantify how much the protein is able to deform the membrane, which is called a spontaneous curvature. So this is one type of experiment which has been used now by different groups, not only us, and you will see an example later. But there is also another interesting assay which was developed by Dimitris Tamou, which is called SLICK. And in this assay, what Dimitris was able to do is able to prepare small liposomes, in this case, which have a diameter which is, let's say, below 200, 300 nanometer down to 20 nanometer diameter. And similarly, is able with this technique to measure the binding affinity of the protein depending on the size of the liposomes. So this is a typical assay which have been developed to quantify how much a protein is able to deform a membrane. So this is about bending, deforming a membrane, but as I mentioned, at the end of the day, if you have a clathrin vesicle, it needs to be detached to, after that, being transported into the cell or into the cell. And at that level, there are two important proteins which are involved in scission. The first one, which is very well studied, is dynamine, which is forming a type of ring, which we can see here. But also actin. And using this tube pulling assay, Aurélien Roux has been able to study how dynamine can be recruited in a curved membrane, mimicking the neck of this bud here. You can see here the dynamine is in green, is recruited on the tube, and you can see this green. And in the presence of GTP, you can see that the dynamine assembly is able to cut the tube. You see that the tube is cut in two pieces at the end of the day. So this kind of assay, I'm not going into the mechanism, but since with this assay, Aurélien and with theoretician has been able to propose a mechanism for fission. So this is for dynamine, which is probably right now one of the most understood process for fission. But in the case of actin assembly at the neck, at the moment, it's a completely open question. And I think that's something which, with this kind of reconstituted system, or let's say built-up system, we hope in the future to be able to understand how actin polymerizing at the neck of this bud, for instance, is able to cut it. In terms of fission, there is also other proteins which are involved in another type of topology. And one of them is called 𝗕𝗂𝗏. So the 𝗕𝗂𝗏 are involved in many processes. So you can see on this scheme the different processes where the 𝗕𝗂𝗏 can be involved. So the most important or most known one is cytokinesis. So when two cells, or when cells are duplicating and at some point they need to be separated, and they form a tiny tubular structure, and this structure needs to be cut at the end, and 𝗕𝗂𝗏 complex, which assemble inside the cytokinesis bridge, are involved in fission. So this is for that part, but also at the plasma membrane. So now, right now, the virus are very, people are very excited about virus for different reasons. But the HIV virus, when it has been replicated into the cell, it needs to be enveloped, and at the last process, the last step of the process before it's leaving the cell, before infecting other cells, is the scission, and this scission is performed by this kind of filament which assemble inside the neck of the bud. And there are many other, let's say, processes. I'm not going to the detail, but what they have usually in common, that they need to recruit this 𝗕𝗂𝗏 complex into the neck of a bud. The bud could be a virus, it could be vesicle like here, but it needs to assemble inside the neck. This is very different from the dynamine, which was assembling at the external part of a bud. Here, this 𝗕𝗂𝗏, they have to assemble inside the neck. So it's why they have been so difficult to study in the past years. And we have contributed also to the process. So we have been recently able to encapsulate this 𝗕𝗂𝗏 protein inside a giant liposome. So this would be a scheme here. The protein are here. And what we have been able to show that if we pull a tube, in fact, this is two components of the complex which are labeled. And you can see here that these components are able to assemble only at the neck of the tube. Not in the tube, not inside the tube, but only at the neck. So 𝗕𝗁𝗺 again also has been interested in this fission process, and recently he published a paper in Cell where he has mimicked the neck of one of these structure. It could be an HIV. So he has used a bead which have been in contact with a vesicle, and eventually has been wrapped by the membrane, and they form a neck. So this would be the bead, this would be the membrane of the vesicle, and this neck. And he has shown that he has worked with this 𝗕𝗂𝗏 protein from yeast, and he has shown that if you have a very complex mixture of this protein, which is here, in this case only, and if you have ATP, you can also induce fission. Otherwise, you have, in many cases, a very almost no cut. So at least he has been able to reconstitute the fission by this very complex system. But so far, I would say we have still a question related to the mechanism, because we don't have, it's not like dynamine, it's still in debate how the fission takes place. And we'll finish on this fission by some also very important process, which now doesn't take place in eukaryotic cell, but in bacteria. And bacteria, at least this bacteria here, at some point also need to divide, and this is 𝗕𝗂𝗏, and need to divide. And at some point, when it's dividing, needs to cut the connection between the two mother and daughter cells. And 𝗕𝗂𝗏𝗶𝗵 has been engaged for many years now to try to mimic and to build up a system which is able to mimic the scission by all the protein which are involved in fission. So some of them are called mean, and they are responsible for the localization of this filament, in this case, which are called 𝗕𝗂𝗃, in the middle, at least at the right place where the cells should divide. So she has been able to reproduce the localization of the at least of the filament in the middle of a structure, which mimic kind of a bacteria. So in this case, this is a macrofabricated structure, which is covered inside by a membrane. And she has been able, as you can see here, to reproduce the localization of these rings in the middle of this structure, which mimic the interior of a bacteria. But as far as I know, she has not, and the other people are working, has not been able to reproduce the scission of a giant vesicle containing this system so far. So it's still a challenge to reproduce in a way this fission of a vesicle or something which mimic a cell in vitro. So this was a protein which are normally cytosolic and bind to membrane by different processes. Of course, there are many proteins which are permanently into the bilayer and cross the bilayer. So they are much more delicate to manipulate because they need to be either in a detergent or in at least something which is mimicking the bilayer, and especially having an hydrophobic part. And over the years, there are at least many proteins now which have been purified and reconstituted in giant vesicle. So it could be ion pumps, like bacteriodopsin, for instance, or some calcium pumps. It could be ion channel, which also transport ions across the membrane. But there is also more complex structure which are able to transport drugs, some channels which are mechanosensitive, some channels which are involved also in the transport of DNA of phages into bacteria. So many complex structure, and even adhesion protein as well have been reconstituted. So I think that at least this is something which is feasible. It's hard work, but it's feasible. And these cartoons somehow reproduce the different techniques which have been used to reconstitute this transmembrane protein in giant liposomes. So at least the first one start with protein which are present in small liposomes, what we call SUV, small liposome, which contain protein. And one technique consists at using the same technique that you use for growing giant vesicle, but starting from small liposomes containing protein instead of pure lipids. And you can use either electroformation or a technique which was developed in a pasture, which is a spontaneous swelling of this liposome, dry liposome on a polymer caching. And you can also use, instead of protein which are reconstituted, you can also use native membrane and purify the native plasma membrane and grow giant liposomes from that. Another technique which is also very promising is because it's a help to keep the directionality of the proteins. Is a direct incorporation. So it consists at preparing GUVs and having the protein in detergent, and by removing the detergent, having direct incorporation of the proteins into the bilayer. In this case, the proteins tends to reconstitute mostly in the same direction. And you have to remove the detergent, of course. And you can also use proteoliposome, and proteoliposome protein are usually much more unidirectional. So if you fuse a small liposome with a giant vesicle, you might end up having the protein reconstituted also in one direction. And you can also fuse native membrane SUVs. So this is different techniques, and I recommend also, if you care about giant vesicle, I recommend a book which has been edited by 𝗕𝘂𝗶𝗹𝗶𝗆, and which was published last year, and which explained everything about giant vesicles. And we have a chapter on the reconstitution of transmembrane proteins in this book. So I wanted to mention also a project which we tried somehow to build up not only a cell a few years ago, but we were very optimistic and we wanted to reconstitute, let's say, an action potential in a system which looked like an axon. And this is a project which we started by 𝗕𝘂𝗶𝗹𝗶𝗆, Sophie Aymon at the time, and Matthias Garten. And our idea was simple. We wanted to reconstitute some voltage-gated channels into a giant vesicle, pulling a tube that we know very well how to do. This will create a structure which looks like a soma and an axon. And we wanted to excite the action potential by a stimulus electrode and to measure with another electrode the signal at the end of the pseudo-axon. So the most difficult part of this project at the end of the day was to induce a transmembrane potential which keeps the vesicle stable. And Gil and Matthias have been working a lot of time, and they managed to successfully have a system. As you can see here, we have a system where here we have the 𝗕𝘂𝗶𝗆, which is still completely isolated from the pipette, which contains an electrode. And at some point here, we have broken with a zip. We have broken the membrane which is here, and you can see in this case that the fluorescent liquid is able to penetrate inside the vesicle. So in this case, we find a protocol to keep the vesicle intact, not growing into the pipette, and we were able to induce a transmembrane potential across the bilayer with this technique. So we illustrated that not with a voltage potassium channel, but we did that with gramicidine. And you can see on this graph here that if you have gramicidine incorporated and you have a case where when the membrane is symmetric, nothing is going on. But if you have outside a buffer which contains choline, which is impermeable to the gramicidine, you create an asymmetry, and the system spontaneously, when it's perfused, is able to create a current. And now, if you apply a potential across the membrane addition of this perfusion, you can see that now you have something which shows that you have an active current, which is now driven by the electrodes through the membrane. So I would say we never succeed to do that with the potassium channel that we were using. So we never succeeded to get this action potential working. So I think this is still a dream for me to be able to do that, but for the moment, it didn't work yet. And I would say one of the issue with this reconstitution is that it requires a lot of biochemistry, and also which is not easy. And you need also to be sure that you preserve the activity of the protein, which is also another issue. And as I mentioned, also in many cases reconstitute the protein, but they could be let's say incorporated in one direction or the other, and a mixture of the two. And there is a very promising system which has been developed by different groups, which now consists into encapsulating a gene expression system, a system which is called PURE. And with that, you don't need to purify the protein. The hope is that the protein is directly produced inside the 𝗕𝘂𝗶𝗆 and eventually incorporated into the membrane of the 𝗕𝘂𝗶𝗆. Okay, so I will finish with cortex, with the actin cytoskeleton. So I mentioned that the hope is also to get a membrane which contains a cytoskeleton as well. So there are different techniques which have been used, and I already mentioned this inverted emulsion, the deep technique. So you can incorporate the constituent of the cortex inside the droplets, and eventually you could form actin layer inside the vesicle, which was done by Cecile Sykes, for instance. And there is, I think, a very nice technique which has been recently developed by 𝗕𝘂𝗶𝗹𝗶𝗆, where he's able to prepare a vesicle which originally is surrounded by a polymer layer, and by using a pico injection, is able to inject into this liposome not only actin monomers and actin-related protein, but also liposomes containing protein. So is able to kind of reconstitute the adesome into this system. So I think this is a very promising technique that he is developing, and we'll see what is going on. And I wanted just to show you some example done by, for instance, Cécile Sykes, where she was able to reconstitute a cortex outside a vesicle. And you can see that she could get with this different protein, different deformation of the membrane, something which is called spikes, and there's a lot of tubes outside. And this is a similar deformation which were observed by Andreas Bauch, but in this case, he encapsulated the system using this C-dice method, and he was also able to see, you might guess, in 3D some protrusion here, or in some cases, some invagination, and also some case where the system is flat. And I wanted to mention some work was done by Cécile Sykes a few years ago, where she also outside, she was able to incorporate as well actin myosin mini filament, and she was able to induce some contraction. And as you can see here, some crack into the cortex or some contraction of the vesicle. So this is a lot of system which should be developed in the future as well. And I will finish with this example. So this is also something that we wanted to reconstitute with Fong Chin-Sai, who was postdoc in my group, and now she has a permanent position in the group together with Pekka Lepalainen. So we wanted to reconstitute generation of this structure, very tiny protrusion, which are formed at the edge of a cell, which are called filopodia. And it's known that this structure, which can grow and shrink, etc., at least the model which was proposed, that they are generated by some IBAR protein, which I already mentioned, this protein which are very and the recruitment of this protein and the clustering of this protein was proposed to induce the recruitment of the protein which recruit actin, so that you can see here. And by recruiting actin, polymerization of actin will induce a force on the membrane locally, and the growth of this filopodia. So we wanted to check if we were in condition and we work with this minimal system, if we would be able to produce localized protrusion. And so what Fong was doing, she used an inverted system. So she put the protein outside of a GUV, and she used the 𝗕𝘂𝗶𝗶𝗆, the IBAR domain, the full-length protein with an IBAR domain, the actin polymerase, which is VASP, and actin, in condition where normally, independently, the protein do not form protrusion. And when she has them together, she was able to form, as you can see, locally, a structure which looks like the filopodia, which contain actin and VASP, and in this case, 𝗕𝘂𝗶𝗆𝗆 is not labeled, so you cannot see it, and you can see that we have a nice also labeling of the membrane. So basically, this is it. So I will finish by at least I think I showed you different examples of what can be done to reconstitute different modules corresponding to some function in the cell membrane. You could see that a lot of it also depends on the purification of constituent proteins, actins, etc. So biochemistry is a very important factor of the success of this kind of approach. You could see that if you want to have many vesicles, you can use all these droplets techniques, but which involve oils, and you can have contamination, which is a drawback. I would say that working with actin-associated proteins and binding them in a realistic way to the membrane is also difficult, because many of the experiments have been done usually by using biological system to bind the actin protein to the membrane, and having a full system is even more challenging. And I would say one of the problems is if you would like to have a very complete plasma membrane with all the complexity, it means that you don't need to have only the asymmetry of the membrane, the integral proteins, or the actin and the binding actin to membrane, but you need to combine all of them. And for instance, it's not easy with all the techniques which have been used to encapsulate, for instance, the actin component. These techniques are usually not compatible with the reconstitution of transmembrane protein. So maybe the 𝗕𝘂𝗶𝗹𝗶𝗆 technique will allow to get at the same time, for instance, integrin and the protein which are related to actin inside the liposome. But so far it's a difficult challenge. And similarly, also to reconstitute transmembrane protein into an asymmetric membrane is a challenge. And I would say right now we have to develop more techniques to overcome all these challenges. And I hope that soon we will be able to get everything at the same time. And in this case, we might hope to better understand how these modules work together to create the function which are seen in cells. So I will finish by thanking my group, my current group, all the former members, because I mean, what I was presenting corresponds to many years of work, and many collaborators as well, and you for attention. And I will probably leave the question, the limitation of the technique for the question afterward. And I'm ready to take questions. If I manage, maybe, let's see, maybe I stop that and I can see, yeah.
Thank you so much. You have a lot of questions in chat.
Yeah, so probably I have to go to the chat.
Let's
see the chat. Where is the chat? I see the
it's
people.
on, should be bottom of your screen.
Bottom, no. Okay. Where are the tips?
If you could read the question before you answer it, that would help people that will be watching this recording later.
I just need to find them. Just at the moment, I don't see them. Maybe I see the people who are there, but where
on
is
the bottom of your screen, when you have participants, share screen button, you should have chat button.
Yes. Okay, I see that. I should see that. No, I see reactions. This is incredible. Sorry for that. Yes, I see that. It's good. Okay, so I start with the first one.
Yep, thank you.
So the first question is, when you model membrane, do you model them as a sphere with curvature and more lipid in the outer leaflet, or do you have to model flat approximation? It's a good question. So it depends. If I care for a curved membrane, usually it depends. In the continuous model, I would say, which are usually used to describe the mechanics of membrane, many of them consider that you have, you don't care, and you don't take into account this difference of number of lipids. I would say this is often completely neglected in a continuous model. If you do a simulation, and of course you have to take care of at least even a coarse grain simulation, you need to take care of this difference. Is it fine? Okay, so maybe I go to the second one.
Yeah, most of the time, the people that asked questions don't unmute themselves to ask follow-up questions, so you can just keep going.
Okay, so in the water oil droplet emission, isn't it that you will have oil residue in the membrane? Yes, this is exactly a point. So I think this is really one of the challenge. So we have evidence using, for instance, this transmembrane potentials. We measure the capacitance of the membrane, prepared with this water in oil droplets, and we show that we have a presence of oil in that. So next question is, can you synthesize membrane nanotube outside of GUV and make them deform membrane from the outside as well as inside? Like, do you need to make or encapsulate the nanotube inside the liposome to work, or can it be added outside? So the nanotube we form are directly continuous with the bilayer. So when we form them, or maybe I'm not sure. If it's a GUV where we form the nanotube, we form them by pulling on the membrane directly. So it's directly continuous. And it's possible also, it's a bit more complex, but you can also do the same thing. Instead of having a bead which is outside on which you pull, you can pull toward the inside and you can form a membrane nanotube also towards the inside. And if it's a membrane tubule which are formed by proteins, you can have them towards the outside, towards the inside, depending on the type of protein you use. So, and if the protein are incorporated inside, they might create inner invagination also, or protrusion, depending on the shape of the protein. So I continue. So the next one is when you make 𝗕𝘂𝗶𝗹𝗶𝗶 filament, what control which part of the membrane they go to? How do they position them in the middle? I think this is, it's not my work. It's a very beautiful work which has been done by Patricia Bassereau, and she showed that this is a kind of a dynamical system where by reaction diffusion of the component of the MIM system, they spontaneously kind of diffuse and react, and in a system which is confined, like the cavity she was using, they spontaneously locate in the middle. So I encourage you to read the papers from Patricia Bassereau's group on this respect, but this is a result of a dynamical system reaction diffusion. So next one is, how do you control direction or insertion for ion channel into artificial membrane? I mean protein expressed inside the liposome. This, I would say, for the moment, I'm not sure that, I'm not sure they will, I think if they insert into the bilayer, they will insert, depending, it's a little bit like they insert into a small liposome. They will have some residue which have a stronger affinity for the bilayer, which will insert first, and then will probably dictate in the direction the insertion of the protein. But I'm not sure that this is something which is very well controlled so far from the pure system into giant liposome. I'm not sure that there was a clear result on that so far. Next one. How do you control the variable pH inside the artificial vesicle? As pH plays a vital role in determining the efficiency of the process it is involved in. I would say that is a very good question, and I would say I didn't think to it yet, so I have no answer to that. So, but yeah, so don't know. I guess you need some special reaction that you might trigger by some optical system, but I'm not sure. I mean, I'm not sure that anybody did that inside a GUV yet. So next one, when you make Filopodia, can the synthetic cell then crawl in certain direction? For the moment, our Filopodia are really are very static, so and there is only Filopodia. So I think for the moment to crawl, you will need to have adhesive molecule, etc. So it would be great, but we are not that advanced yet. So maybe if you come back in 5 years, maybe we are able to do it, or somebody is faster than us and does it before. I hope. So for the moment we can't, but it would be very elegant to do it, but we can't. Okay, I continue. In considering the incompatibility of these modules, it might be useful to consider the origin of life. Do we know anything about the order in which evolution made these modules and brought them together? I would say, I'm not sure I'm the right person to answer that, but I would say to start anyway, at the beginning, there was probably you need to start with people try to understand how you create a membrane from scratch from the element which created the first lipid or something which can create a compartment. So I would say people have already a hard time to understand how you create a bilayer, or at least you make a compartment from scratch. You could see that also in the archaea system, they need at some point to reproduce and to fission. And this arcade system, they all have an escort system. So it seems that the escort system, the 𝗕𝘂𝗃, which might be also a little bit similar, seems to have been there very early in the system to induce fission. So I think that this is a key problem. I would say the bar domain I was describing, this is something which comes probably later for much more advanced and regulated processes. But I would say the 𝗕𝘂𝗃 and the escort, which have been involved in the fission of at least this type of filament, are very key for if you want to understand fission. So I think this is why it's so important, I would say, to understand that at the moment. Next one is not a question, so I don't read it. Next one is, can you generate and maintain membrane potential in artificial system? Yes, with our electrode, in principle, we could keep the transmembrane potential for at least half an hour without any problem. So I think that this is very promising. And I think that could be used more. I mean, I think not many people have been using our system yet, but I think it's a very good system to keep transmembrane potential constant across a barrier. Next one is, this is a paper on 𝗕𝘂𝗃. So I think this, thank you very much, Anna, for the person who was asking how 𝗕𝘂𝗃 might find a division site. So there is a reference that you could find in the chat. And next one is, if you add halogen atom to any protein, will that enter into artificial system, meaning chlor and sodium, etc.? I would say I don't understand much the question. So I don't know. I don't absolutely know how to answer this question. So I'm sorry, there was the last one, but I'm not able to answer that properly. So if there is nothing else?
No, I think you answered all the questions in chat.
Okay.
Thank you so much.
Okay, thank you all for your attention, for your questions, and I hope that I give you some motivation to try to use some artificial membrane to address some question in the future. So if you need any help or anything, don't hesitate to contact my group, and we'll be happy to help if we can. But there are many other groups, and I hope I didn't mention all the groups who are working on membrane in the world, but at least I try to mention most of the group who are developing techniques, etc. So you could have an idea about where I knock on the door. Okay, so
thank you.
Have a good day and a good evening for those in Europe, and I wish you the best.
Thank you so much, and bye-bye.